Welcome to the Qualitalks podcast, a show about pharma and GMP. The pharmaceutical industry is a fascinating and complex field, and it takes countless people to bring a product to the market. In this podcast, we bring you some of the industry's brightest minds who will share their wisdom with you. And now, please welcome your host, Jan Kugel.
Welcome to the Qualitalks podcast. I'm Jan Kugel, and today we are talking about Process Analytical Technology, or PAT in short, and why it's no longer just about measurement. For years, PAT was treated as a technical add-on, sensors, spectra, feasibility studies. But if you look at where the industry is today, that view is outdated. PAT has become a strategic tool for building quality into processes, reducing uncertainty, and moving from testing outcomes to understanding and control. And my guest today is Stanislav Kazakov. He holds a PhD in chemistry and brings more than 10 years of hands-on experience across PAT development, implementation, and scale-up. Currently, Stanislav is a senior business development manager at Thermo Fisher Scientific. As you might already know, Thermo Fisher Scientific supports pharma and biotech teams across the molecule journey with integrated analytical solutions that advance discovery, scale manufacturing, and ensure quality and compliance. And in this episode, we'll keep things practical. We'll cover the PAT journey from niche to strategic use, real examples across academia, chemicals, and pharma, lab to plant transfer, common obstacles, and where sensors and AI are taking us next. So with that, let's get started. So Stanislav, welcome to the show. It's great to have you here. Very excited to be talking to you about this topic.
Yeah, good morning, Jan, and thanks for the invitation. Very exciting to be on your podcast.
Thank you, Stanislav. So it is, I believe, the first time that we are talking about PAT in our podcast. We have been talking a lot about quality and manufacturing here, but PAT is also a very, very important part right now in the manufacturing, and it's very exciting to dive in to it with you. So before we start with PAT and talking about that, maybe you could give us a short background about yourself and how you fit into all of this.
Yeah, thanks, Jan. So I studied chemistry because I have a very curious mind, and within the chemistry, I then started to study catalysis research, and within this catalytical research, I did a lot of analytical. I used a lot of analytical tools to order to understand the kinetics and the mechanistic aspects of those processes. And this is actually how I end up in the PAT field, is because I was really curious to understand what is happening, and I used a variety of different analytical tools, and this really helped me to understand, how to improve, first of all, the synthesis of catalysts, but also overall how to improve the processes. And with the PhD in chemistry, I then stepped up and became the PAT scientist, and I'm doing this now for 10 years. And it's really exciting. It's really great to be part of this journey, and look forward to discuss with you some of the PAT topics today.
Excellent. So, Stanislav, in your own words, how would you describe what PAT is and its purpose from your perspectives from academia to the industry? What does it mean for you?
So overall, I would say PAT is the sum of all information we need to characterize, understand, and develop a process from laboratory into scale. So basically, people, researchers, they need a certain amount of information in order to deliver a molecule, a product into the market. And along this journey, they need to get this information in order to be successful. And PAT is, of course, now essential part of getting as much information as possible in order to characterize the processes in depth. And I think this is a really broad definition, but I think this is a really good definition when it comes to what do we need PAT for. I think overall, PAT was a niche application. It was actually developed to characterize difficult to probe chemistries such as isocyanide chemistries. And now it's become a really essential tool for the high value chemical, but also for the biopharma industry to characterize processes in real time and make the researchers and scientists successful in driving either new molecules into the market or driving the recipes from laboratory into scale.
Right. So what you're saying that it started as a niche technology, right?
Mm-hmm.
So through your about 10 years in the industry, but also before in the academia, how do you see this technology changes and develop? So let's say you started in the industry like 10 years ago, right?
Mm-hmm.
So what has changed in terms of the technology itself, the mindset, how people accept it or ready to
Mm-hmm.
accept it in the industry? Has something changed? How could
Yeah,
you
there is a
describe it?
lot of changed, and I think it's not only in 10 years. I think if you think 40 years ago, it was quite big machinery you had to maintain, and you had to be really a specialist in optics and spectroscopy, but also you need to be able to operate these difficult to handle, difficult to operate machineries. I think the new technologies which are on the market, first of all, miniaturizes the equipment, so make it small, make it available for everybody to use at the same time. It's also not all. It's easier to implement into processes because we, yeah, it used to be that you need to take the sample into the quality control laboratory, and analyze the sample. But now, because the instruments became smaller and you're also able to couple the instruments with fibers, you can now bring the instrument into the process, into the sample. So overall, I think when I look back, it was a very tedious technology, and now it sits on the bench. It's easy to operate as a balance or a pH electrode. So basically, two things happen: the materialization, technology advancement, but also the user interface. So the software has become more adaptable and more easy to use for chemistries, for process engineers. So overall, I think the market drives the technology because it's really a really great tool to use. At the same time, we have now the capability to use it from the bench, and everybody is capable of using it, so to say. So overall, yeah, when I look back, a lot of things has happened, and it's not only that the technology was driving it, but also the market and the market needs drove the development, utilization, and the use of those technologies. Yeah.
Mhm. Right. So I've seen it mostly during my time in the industry. The PAT used in biotech, and
Hmm.
it was in a new facility. But before that, I rarely seen it around. So would you say it's applicable not only for biotech but for all kinds of molecules, small molecules, big molecules, and so on? Or you would say there are some application where it's uh, more needed than in other applications?
Yeah, it's a very interesting question. So, as I already described, it was really like developed basically from niche, chemistry, small molecule worlds, and basically what it allowed everybody to put like a sensor and to have like an insight of what is happening within the reaction, within the process. And because it's a tool, you can use it anywhere. You can use it for academic research. You can use it in chemistry departments. You can use it in biotech departments. And I've seen various different of applications. And I think this is what is really exciting about this technology is you get to see a lot of different applications. You get to, also different touch points with various researchers. along the way. So we have we have our core, let's say, core application areas, as you mentioned, the biopharma, of course, the pharmaceutical industry, the high value chemical industry, the academic industry, but there are also some emerging applications along that. So if we talk about oil and gas, if we talk about the green energy and the green resources, where people want to understand how to optimize it, how to make it cheaper, better, faster. And I think this is also the beauty of PET, is that it's not has like a distinct application field, but you can do so many different things with that. And I think this is also what drove me into the field, is my curiosity to learn more about not only the chemistry side of things, the technology side of things, but also what else can you do? What else can you probe with your PT methods in order to understand, characterize? And I think this is also one of the key message. Innovation starts with understanding, and PET is a core essential part of this understanding. So basically, you have a black box, there is a process going on, and now you're bringing a light into the process, so to say. And you enlightened the scientist, you enlightened the researcher, because now you can see what is happening in real time, what is happening under reaction conditions. And once you have this understanding, you can now convert this understanding into what are the critical process parameters, and how these process parameters influence now the quality attributes, for example. This sort of like level of understanding really allows you to develop a robust and reliable process. And this is really the key now factor to drive molecules successfully into the market. So back to your answer, back to your question, the answer is it's applicable everywhere. And this is also the beauty of PET.
It seems very critical and helps really, as you say, understand the process. So would you say it's mostly important and applicable for process development, the validation of new processes? Or would you say it's also right now that it's so available and easy to operate, that it's also helpful for existing processes to support quality control and so on? Or would you say it's mostly for the validation, the development of new processes? How do you see it?
Yeah. When you look at the molecule's journey from R&D all the way to manufacturing, it all starts at you need to identify a molecule in pharma industry which has a therapeutic response to a certain disease. So scientists are screening through thousands and millions of different candidates to find this one specific molecule which has a certain effect. Yeah. Once they found this molecule, usually the R&D scientist is giving this molecule to a chemist, to an organic chemist, who is then in charge to find a recipe to synthesize this molecule from base chemicals, right? And usually you're using a retrosynthetic approach to define the reaction route, and to find what are the best possible reaction conditions to end up having the desired molecule. Once you have this molecule, you then go and talk to the process engineer and say, look, I have now this recipe designed in the laboratory. I want to scale it into a larger manufacturing plant. And now the process chemist and the engineer needs to translate this recipe in the laboratory, which sometimes works in the laboratory, but not always translates into scale. And this process engineer needs to find, a robust and reliable recipe to scale it all the way into manufacturing. And this is a very tedious task to do. And then in the end, of course, you need to manufacture this molecule with all the different quality attributes in place under the regulated environment. So back to your question, where do we need PET? My approach is always as early as possible. Yeah. So basically, what we want to do is we want the organic chemist to start using PET, so he can actually look at his process or his reaction in lab scale and already have figured out what are the critical process parameters and how can I optimize my process in a laboratory scale. Because if you do start early on, this helps you really elaborate a good strategy in order to be successful during the scale. So traditionally, it was really used as a sort of like troubleshooting device. So, for example, as I mentioned, difficult to monitor chemistries or something is really not easy to understand. You would go ahead and put PT in place to give you the information you need. Yeah, and now, and I think this is also related to the whole data automation digitalization stream which we see now. It's not a matter of troubleshooting. Now it's a matter of, hey, I want to have as much data as possible. So if I start already at the earlier stage, I can use now this data later on when something is needed to be looked at or something is happening. Now I have all the data information there to rely on. So and I think this is also the shift in the trend where it was only used as a troubleshooting device, going from, hey, something is not working in manufacturing, we need to scale down the process, and the process development chemists need to start to look at. Now towards the organic chemist is already using PET. He has developed a method, and now we can drive this method all the way from the bench to the scale. And I think if companies are using the right approach to start early on, they can also easily implement PET in all the different other steps as well. And I think this was also a little bit, the downside. If you are a little bit too late with PET, if you are already in a stage where you manufacture the molecule and now you need to use PET, you will never be able to then bring PET into its natural habitat, the process environment, so to say. So I would answer the question, use PET as early as possible because it provides a lot of information, a lot of data density information, which is always useful when scaling chemistries, biopharmaceutical recipes from smaller to larger scale. And this is applicable not only for the drug substance manufacturing, but also for the drug product manufacturing in pharma. And as well as if we talk about biopharma, we have the two different houses, so to say, the upstream and the downstream, where PET can be really embedded in most of the process steps. And I think this is also what we see, this shift in the thinking of, hey, this is a niche technology, specific use cases. We need to have a strategic investment on PET in order to have as many steps as possible driven through PET, well characterized. So later on, we have a very well developed process to rely on.
Right, Stanislav. So what's interesting for me, you mentioned that you should start PAT as soon as possible. And it's more difficult to implement it later on.
Mhm.
So can you actually bring PAT to existing manufacturing processes and commercial product that already runs commercially for years? And does it make sense?
Yeah, it's a very good question, Jan. Yeah, because once in pharmaceutical environment, once something has been filed, it's then of course a question is, yeah, how easy can you make adjustments to the process? Usually, it's then you need a business case scenario where you need to identify, why is it better now to use PET versus the traditional filed method? And I think this is also one of the problems associated with PET, is that if you are only starting doing PET when everything is validated and everything is filed, you will never have so much manpower and such a huge business case in order to change the already filed documentation. This is, I think, what I've seen in my experience is that you will hit a wall and you cannot just overcome certain hurdles. Because there are many more hurdles to come. So I think this regulatory aspect is like a final wall, which if it hits you really hard. So I think it's better to be prepared
Right.
for
So
that.
if you miss the train, it's then very difficult to jump back on. Uh,
Hmm.
but so I can consider. So I think that although uh, all processes are validated when they come and submitted, and uh, companies start manufacturing the drugs, but I guess there are still many processes that sometimes I don't know. Let's think about the corona vaccine, right?
Hmm.
They needed really quickly to start manufacturing a drug that hardly been tested on, and
Hmm.
it was a real challenge for the industry, right? And I believe when you create such drugs or such processes to address really quickly some problems, or to release some drugs that should help people, like cancer drugs that sometimes are allowed to be released quicker because they uh, for they're life saving, so it's okay to have more side effects and so on, right? Uh,
Hmm.
but I think that such drugs that are quickened to the market, probably the process shows much more deviations during the process. You always find out that yeah, so I presume that more deviations come up in the process because it was validated, but I guess if you do it with PET, you probably get less deviation in comparison to uh, products that
Hmm.
didn't use the PET
Yeah.
to really correct it. So the question is, there are processes where there are so many deviations on the line and so many batches are thrown away, whether it
Hmm.
makes sense to go back to the drawing table and still bend
Hmm.
regulatory. What's your perspective?
Yeah, yeah. I mean, yeah, it's a great assumption, and to say, look, if I embed the quality into the process rather into the end product, of course, then I have a better control. Yeah, and this is also what the FDA back 2006. Yeah, they made a framework in order to really push PET towards where it needs to be. So PET now is more or less used in order to characterize processes in order to better understand. But of course, in the end, what we want to do is we want to use this real-time information to have a better control of a process. And, we need a system to do that. And PET is the system to do that, of course. Now, how often do we see now PET in manufacturing? Since 2006, since we have this framework, and FDA is now pushing PET quite a lot, and companies are investing. We see that it can be used in order to drive, of course, molecules fast into the market, to have better quality of the product, and this is, I think, overall what the pharmaceutical industry is obliged to. And you're right to assume, if of course you have real-time insights, you can better control the quality of your product. And not only that, you can also drive molecules in a more designed and designated way into the market. So of course, everything is connected together. Now, I think, it's also not a straightforward way to do that, because you need people who are driving those strategies. You need people who know the technology. You need everybody to be on board. And I think this is like more a cultural thing. When we see also a cultural shift in the industry, is that it's not a question of we like to do it or not. It's a question, how do we do that? Yeah. So I think your question is correctly placed. Now, you also said, if we have more PET into place, do we have less deviations? Yeah, and I think this is something which is non-dependent on the technology itself. It's only dependent how robust and reliable is your process, and how can you deal with certain aspects during the manufacturing. Yeah? So there was a very interesting saying: if something can go wrong in manufacturing, it will. So if you want, you need to have, of course, a safe environment to operate, safe and robust processes to scale, and also you need all certain risk assessments and strategies to be applicable, when something appears and something goes wrong. So now we go towards this golden batch trajectory, and we can always monitor what is happening in real time. We can also react in real time, and this is, I think, overall where the journey is going. So the journey is going, towards a strategic implementation of PET as soon as possible to drive these methods into manufacturing and to really use those methods for real-time control to ensure that you have a consistent and reliable quality of your product.
Right, right. So thanks for this perspective. And so you mentioned quality attributes and critical process parameters. So maybe we can connect it also to what we have been discussing. Maybe if we go one step back and maybe say a few words about those terms a bit and why those are important to help operators and scientists embed quality into the product. And also, you said that correctly, the PAT helps identify those quality attributes more precisely and assess the critical process parameters. So would you say also that without PAT, you could miss some of the critical parameters, and then it impacts also the quality as well? So I know I asked quite.
No, I think it's a good question. Yeah. So when you look at traditional methods, yeah, what are the traditional methods? Usually, people take a sample, and they take a sample and they characterize the sample, different methods, and usually offline analytical methods, and they say, okay, this is how the sample looks like, and this is the chemical composition of the sample at a given time. And it worked, more or less in a good manner. Now the question is then always, how often do you pour a sample? Usually when somebody is around, when you have the capability to do so, and sometimes before the reaction starts, maybe you analyze the raw material identification, and after the reaction finishes. And basically what you have is you have a process which runs for a certain period of time, but you don't really understand what is happening in between. And I think this is where it's you miss a lot of information of how the process behaves and what are the different things in order to drive from starting material towards the end. Now, what PT is now capable of doing is, first of all, you have a higher density of information. So instead of like pulling a sample, let's say every 4 to 6 hours, because this is usually what is done in industry, or in the biopharma industry, maybe once a day. Now you're capable of having an insight, let's say every minute. Yeah. So you have every minute you understand the chemical composition, and you also usually monitor the physical parameters such as temperature, agitator speed, pH, maybe oxygen, some other univariate methods. And this information now you can link together. So you can link together the process parameters to the concentration parameters, and PT is adding like an additional dimension into your process by giving you access to a high density of information. And this high density information now allows you to understand, like, okay, so if the reaction temperature increases, or if my pH level drops, or my oxygen saturation changes, how does this influence the quality or the concentration composition within my process? And this little insights allows you really to understand, how can I now operate in a reliable manner, because you have now linked the process parameters to the concentration parameters, and this gives you really an insights to when everything goes right, how it should look like. When something deviates, what is actually happening. So I think this, I would say, like the PET is a third dimension, connects the correlations together and provides you the possibility to understand this and later on also act upon this. So you have now different scenarios where you say, okay, if I have let's say something happens, my reactor starts to heat up, there is a temperature build up. How does this influence my chemical composition? And later, actually, if you invest a lot of time during the scale up, and this is also what I've seen so far, you have such a well understanding of your process, because it's so well characterized. Now you can actually go in manufacturing and use maybe just a temperature or pressure sensor, because this allows you always to understand what is happening. So PT that doesn't always necessarily translate into, we have to have, as many sensors as possible in manufacturing. PT actually allows you to get this process understanding on a such deep level that maybe later you don't need PT, because you characterized and understood your process in a such detailed way.
Right. And can you use PT also for in-process control? So you said it captures the information very quickly. So we go a bit back to, because I asked you if it can help also support quality control besides the no process development. But if
Mhm.
runs continues running when the process is already validated and
Yeah.
you continue looking at the process, so can you use it as well in parallel as additional dimension to quality control?
Absolutely. And I think this is also what's been used in the industry already. So now, instead of relying on quality towards the end of manufacturing, people are using PET, and they embed the quality into the process and have it as a go, no go scenario, or as you said, in process control. So basically, you can also do real time release testing based on PET. And this is also what we've been doing. I mean, we're doing it, of course, in-house, but we also are using this technology to create, experience, knowledge, to bring it to customers. And we do it in different ways. Yeah? So PET is not always a sensor which sits in your process. Sometimes it can be also at-line technology, which you can use as early as possible in order to have, a quick line into the market, meaning that as soon as a product is manufacturing, you can already analyze it and release it into the market. So it goes both ways. Yeah, you can use it as real time control. You can use it as at-line real time release testing, and of course companies already doing that. So it's not something which the future will bring, but it's already running in manufacturing.
Yeah, because it just makes sense if the technology is already developed to such um, a stage where you have the right software and everything just makes sense, to not only rely on the fermenter parameters itself, the internal ones, but also the additional layer. And um, how would you say the technology is right now like developing? So does it did it reach the peak, let's say, in terms of what it covers, or is there still development in uh, trying to be able to analyze additional um, things and molecules? So let's say viral vectors now are being used, or
Mhm.
mRNA. So is the technology already there to cover all those types of modern products, or is there still a
Mhm.
development or customization needed for each
Mhm.
client, for example?
Yeah, yeah. I think there are two different things. I think first of all, using PET is it still requires a PET scientist, and what we need to develop now is our tools to really replace a PET scientist. So making sure that PET, because usually it's a spectroscopic technique, and now you need to turn spectra into concentration, and concentration into data, and data into actions. And you still need to do certain things like chemometrics. You need to build models. So I think the development of this machine learning algorithms in order to really replace a PET scientist and make a Raman probe work like a pH probe. So basically, you just using a probe and getting real-time data, and you can act based on this data. So this is one development which we currently see, and there are now companies who really help to really bridge this, let's say, PET scientist gap. And of course, going from this really difficult to analyze chemistries from isocyanates in the early 60s, 70s, now we have new commodities, we have new molecules. PET is highly driven through the biopharmaceutical market because, of course, the biopharma reactor, or the biological reactor, you have living cells, living organisms. So it's really important now to have a sensor and watching what is actually happening under those conditions, because every reaction runs slightly different, right? So we have established PET for monoclonal antibodies, for also other new modalities. We have also great literature when it comes to mRNA vaccines. So it's all out there, and it's already applicable. Now, when it comes to new modalities, so gene and cell therapies, this is something which is still in development, because these are really, single batches which are engineered based on a human genome. So it's a personalized medicine. It's everybody is interested into doing that, but because it's a really new commodity, new molecule, it has not been still established yet. But I think everybody is interested because, of course, here again we have we need insights, we need understanding. Does it work? Is it working the way it should be? And so on. Do I have my quality attributes in place? So I think it's quite also exciting, because the technology itself is applicable to all the different commodities, and it's really exciting as on the way to monitor what is happening. And we, of course, also have a high interest to establish PET into this new commodity, cell and gene therapy. So this is something which is on the radar, currently ongoing, and as soon as there will be some news, I can let you know.
Okay, good to hear that
Yeah.
developments are still in the move. And if we go a bit to the micro of the process itself, right? So it starts usually in the lab, and then it's scaled up uh, in the manufacturing plant. So I know that sometimes things go wrong, right? So sometimes it happens that you do things in the small scale, everything goes well. You
Mhm.
scale it up, and things start to break, right? So it happens, right?
It happens. Yeah, it happens a lot. Yeah, I think you're right. So something which happens, which works in the laboratory, doesn't translate into a larger scale, and this has to do with all certain phenomena. And I think this is also what where PET has established itself. Because now you're able to um, get the data density from laboratory into scale, and connect the process parameters to the quality attributes. Now you can really define how the recipe should look like in the larger scale. And this was, so to say, the go-to method for the last 10 years. PET was used in order to bring those recipes into scale. And you mentioned that something goes wrong. I think it happens quite a lot, yeah, because this is in chemistry world and in biopharma world, where we're all dealing with things which are sometimes non-tangible, or maybe we do something which we don't fully understand. So PET is, again, I really like this, to bringing the light into the black box, where you can see, hey, how what are the different things I need to do in order to be successful. And PET is now essential part of not only bringing this recipe into scale, but ensuring that you have a well characterized process, a well understood parameters. So if something goes wrong, as you said, you also understand what to do in order to make it right. Yeah. So later now, of course, it's not only used to that, but also now to control your recipes in real time, to also understand, hey, I need to deliver certain quality of my molecule because it goes into the patient. And now I have the tools in order to monitor not only the end product, the pharmaceutical molecule, but also I have the tool to monitor my process and in order to control the process so I get the right quality at the right time.
Right. And if you would or could pinpoint one or two most important things to get right when you move from the lab to the plant and scale up, what would be one of the most important things to get right and not to do some mistakes that are common when moving to the scale with PAT?
That's a that's a great question. What is the most essential in order to get things right? I think most essential is, of course, to be curious, to say, look, I want to. First of all, I need to make sure that I characterize my process in a way that I understand it. I need to make sure that I communicate my findings, because the language of an organic chemist is different than the language of a biological chemist, a biochemist, a bioengineer, or a chemical engineer. So I think communication is essential when going from R&D all the way to manufacturing. So people need to talk to each other. And this is also, I saw myself as a PT scientist, I was really connecting then all the different departments together. I was, connecting the lab scientists to the process engineers, to the quality control scientists, to the QA, so quality assurance, all the way to the operators and engineers. So in a way, in order to be successful, and you can only be successful as a team, it's really essential to talk to each other, to share findings. And I think this is the beauty of PETs, because you have now scientific data and proof. Now you can really talk about the science behind your process, and you can now say, look, this is what I've observed, and we need to do that based on the scientific findings from my team, from my sensors. And I think this is really essential in order to be successful, is to work together. We have different type of brandings in the pharmaceutical industries. Some call it CMC, chemistry manufacturing, or MSAT, and there are so many different roles. I think in the end, it's really important, and I think it comes maybe to the company culture, to talk together, to work together, and to use, of course, data as the foundation to scale those chemistries successfully. I mean, there's always something happening, and it cannot be always right. But I think the pressure industry to get things right the first time is really high, and as we also see, the chemistries or the molecules are getting more and more complex. We used to manufacture small molecules. Now we're talking about biological molecules, monoclonal antibodies. Now we're going into gene and cell therapy. So the level of information is steadily increasing. And of course, in industry, everybody wants to be faster to the market. So what I've seen is that this information rate increase also influences how team works together, and what tools do they use in order to be successful along this journey.
Yeah, thanks for that as well. And I must ask you something that I must ask, right? I think right now it's like all everywhere. A question about AI. So everybody is in an AI race at the moment, and
Yes.
I'm sure that the PAT is no different. And what is the status right now? So I'm sure Thermo Fisher Scientific, they're also part of the race. They're probably doing something to implement AI in different products they sell and services, and I'm sure that PAT is part of it. Probably is a part of trying quickly to analyze, to understand results. So that's what I could think. You get a lot of data, so probably AI can help you sort the data quicker, get the results. So that's from my opinion. But what is it on the
Absolutely.
ground from your
Yeah.
perspective?
Yeah, AI, of course, is more or less essential to in order to drive P&T where it should be. And there are certain things, and we also, of course, develop. We have heavily invested together with OpenAI in order to embed, of course, this technology into our solutions, into our products. But I think overall, AI is going to replace the PT scientist at one point, and should replace it, yeah, because in order to get insights from spectroscopic data, you need to build chemometric models. And of course, some of these chemometric models already use AI, but it's also towards the validated chemometric models. And there is a lot of stuff happening. And when you go to conferences, and I go to conferences, I'm always very interested, keen to understand what is happening out there. And I've seen a really insightful talk about AI agents. So instead of using a technology now, you can use AI agents in order to just tell them, hey, this is what I am doing right now. This is what I want to have having in the end, in data, decisions, real-time control release. How do I do that? And now, in the AI agents are capable of giving you a path in order to get there. So there is a lot of stuff happening, and it's really exciting because PET is the foundation. Then it gives you the data, and together with the data, now you can define where do you want to go, what are the things you want to achieve with this data, and without having a lot of mathematical knowledge or statistical understanding or chemometric building, you can use this generative tool in order to really gap from spectra into real-time insights, into real-time decisions. And this is the key to go. So in the end, AI will and should replace a PT specialist, so that everybody can use PET. AI is really essential into driving PET into the real world applications. And we are also part of this journey. So we are also investing a lot to be part of this journey, to be successful. And it's also really exciting because now you always need to convince people and to teach people how to use those tools, how to clean the data, how to now analyze the data, how to build chemometric models, how to get now insights, and how to use these insights and data for future. And now this is something which AI can take over. So AI can teach you what to do, or just build you something which you can just use. Of course, in the end, you need somebody who understands this in order to sign off and in order to double check if everything goes right. But I think AI is a great catalysis or catalyst, which really brings PT into everybody's world.
Right, good to hear that we are going into this direction of automation, quicker data, but also now we also understand who should polish their CVs. But but probably we know that AI will replace a lot, but all those people that their job is being maybe replaced, but some tool, they are not forgotten, right? They will have a different role of double
Absolutely.
checking, analyzing, or I don't know, says what would be then the role of a PhD scientist that now AI does most of its work. It probably will be more of an oversight or developing human input. What would it be then?
Yeah, I think a PT scientist overall is somebody who connects things together and people together and teams together. Because in the end, you need somebody who understands this, of course. But maybe it will be like project coordinator. Because in the end, you need a huge team to drive PET, and a PET specialist will have a different role or perspective in the manufacturing environment, in the pharma environment. So I think overall, when I was saying replacing, I was not meaning replacing the job, but just refocusing on where you should put the effort. And of course, if you take out the effort of this mathematic modeling, then you need somebody who can deal with data integration and driving strategies and making sure that all teams work together and everything is aligned. So I don't want to take people's jobs away through implementing AI. I just want to really bring technology fast into the market so you can use it in all different departments without specialty knowledge, and also use it and adopt it on a broader context. Yeah. So as I said, bring it as soon as possible, scale these methods into manufacturing, and make sure that you are communicating well and working well together. In the end, you always have people working together and the machinery which supports you just do things better, more efficient, and more productive.
Right. So a very good point, right? So it's the jobs are shifting to more human aspects, basically. So the machine can do more the mathematical analysis and so on, and the planning and the algorithms, and the person now needs to have more people skills, managerial skills, bringing teams together and managing the projects. You still need to have understanding of chemistry, but also probably get more hands-on experience or more theoretical experience also on managing projects, products, teams, right? So that's where it's
Absolutely.
going.
You put it well together, Jan. Yeah, it's a great summary of what I just described. Yeah.
Yeah. Yeah, excellent, Stanislav. So fascinating topic, which we probably could keep speaking for a couple of more hours, but I unfortunately need to let you go to your day in business. And as my last question, if there is something that I didn't ask you, but you feel it's very important to share, is there anything that you would like to add before we finalize this session?
Yeah, in the end, I always say stay curious, stay foolish. So basically, I think this was my main driver to adopting PET, and I'm also very happy to see now PET in a better place than it was when I started. So I think it's great to be part of this journey, and everybody who will want to be part of this journey, I'm happy to onboard everybody. So yeah, be the change you want to be, and stay curious.
Yeah, so those are a great encouragement here. Maybe one question to
Mhm.
from my side still. So, if you're looking at PT 5-10 years ahead,
Mhm.
um, do you have any where is this technology is moving besides the AI? Is there anything else? Do you think that is coming that will
Yeah,
change? How it's done right now, or
yeah.
what application?
So I mean, in the beginning, we talked about miniaturization. So the products are getting smaller and smaller and easier to use. So that means in 5 to 10 years now, we will have really easy to use sensors, as well as I think everybody's monitoring now our own health through smartwatches. So I think these also PT measurements can find place in all different kinds of aspects in your daily life, and this is also quite something interesting to think about. We have now the possibility to not only track our heart rate and our sports activities, but I think overall PT will be applicable not only for molecules but also to the human body, so to say. So we can really also nicely monitor the health conditions. So it will be like preventative PET. So instead of producing maybe the molecules which we later on need if we are sick, we can now use PET to monitor our health and to prevent any conditions in the future. So I think this is what I've seen so far, and it was also quite interesting to see what is happening on the market regarding this health trackers and health devices. But maybe it's now 10 years time frame is every company is invested in PET. Everything is run on digital automation platforms. Everything communicates together. So I think this autonomous laboratories, which we also see just making also the R&D a little bit better in a way that it's really difficult to manufacture or to find the right molecule to target a specific disease. It takes 7 to 9 years, and now we can use AI, we can use all different sensors in order to have a lot of different data. And it's not maybe quality data, it's like just all quantity of data, which then in the end gives you a lot of insights of what the molecules are the right one to choose, and maybe we can now shorten the development times of medicines. I think, of course, everybody wants to have a molecule targeting chronic diseases, a molecule targeting cancer diseases. So I think maybe the PET devices will just help us to get a better, healthier, better life overall through monitoring the health and making sure we have medicine in place if something goes wrong.
So it's very fascinating we're heading. So thank you for those insights from your end. So I didn't think about this part of actually taking this process analytics technology and putting it into already live tracking also in people as well, which of course when you scale it down and can catch smaller molecules, you can use it as well in the biotracking of people. So that's very fascinating area we're entering in the era.
Mm-hmm.
Yes, so Stanislav, thank you very much for coming to the show, speaking to me about this.
Yeah, thank you, Jan. It was a really interesting conversation.
Thanks for listening to the Qualitalks podcast. If you enjoyed the show, please leave us a 5-star review. If you want to learn more about GMP, please visit us at www.qualistery.com. Stay compliant and see you at the next one.